Everyone to the second web lecture organized by ISCB-SC RSG-India Node. Today we are honored to host Dr. Sainitin Donakonda, Research Group Leader from Institute of Molecular Immunology and Technical University of Munich, Germany. Welcome, sir. Before we commence the event, I would like to brief about the organization that is ISCB-SC, which is the International Society for Computational Biology, Regional Student Group India. This is your local window to global opportunities. ISCB-SC is a global organization based in the USA. It drives computational biology forward through research training, outreach, and community building. The ISCB Student Council, started in 2006, nurtures the next generation of computational biologists, linking local talent to global opportunities. RSG India connects and promotes a strong nationwide network of computational biologists. There are over 26 countries that are being hosted under the regional student group in ISCB-SC, India being one of them. With regards to RSG India, we proudly collaborate with premier institutions including Mahindra University, NIT Calicut, Mahe and Srihar, which is in Tamil Nadu, which is where we are from. Here is the RSG India Core Committee 2025 to 2026, supported by faculty advisors and visionary leaders across India. RSG India has successfully organized impactful events in the past, fostering collaboration, learning, and innovation in computational biology. Here are pictures from some of the online and offline events organized from 2023 to 2025. You can reach us through our website or connect with us on social media platforms mentioned here. Thank you for listening. And over to Abdul Aziz, Secretary of the Tamil Nadu Node, to brief about the student initiatives that we are able to do from Sri Ramachandra University in Chennai. Abdul, over to you.
Thank you, Harshidi. Good evening, everyone. I'm Syed Abdul Aziz, and I'm the Secretary of the ISCB RSG India Tamil Nadu Node. First of all, I'd like to take a moment to introduce us, the Srihar node of ISCB RSG Tamil Nadu, India, Tamil Nadu. We are a student-led community with a goal to create a platform for young aspiring students of computational biology and bioinformatics, so to collaborate with researchers and industry professionals through various events and workshops and webinars such as this one. And thus, our goal is to encourage learning and collaboration for students and researchers alike. So next, I'd like to introduce our core team behind this effort, consisting of members across the undergrad and postgrad level of Bioinformatics department at Srihar. And a special mention to our advisory board, consisting of our faculty, Dr. Dickie Davis, Dr. Abhinand, and Dr. Arundhati ma'am, who we are very grateful for their advice. And then, without further ado, I'd like to pass it on to Dr. Abhinand to introduce our guest speaker, Dr. Sainitin. Over to you, sir.
Thank you, Harshidi and Abdul. Thank you for this opportunity to introduce star speaker of the day, Dr. Sainitin Donakonda. Am I audible?
Yes, sir.
Yes, sir. Please proceed.
Oh, yeah. So, firstly, before proceeding, I would like to thank Dr. Sainitin Donakonda. I'm very proud to tell that he is my senior. He graduated from our Department of Bioinformatics at Sri Ramachandra Institute of Higher Education and Research back in 2007. He's a couple of years senior to me. And sir accepted to give this talk at a very, very short notice. I had approached him about 2 weeks ago and he readily accepted amidst all his busy schedule. Thank you so much for that. So to introduce the speaker formally, so ladies and gentlemen, today we have a pioneering scientist who is doing some tremendous work in the areas of computational biology, immunology, and translational medicine. Dr. Sainitin Donakonda has dedicated his career to decoding some of humanity's most challenging diseases by using his computational approach. He received his master's in bioinformatics from our university back in 2007, and then he went on to start his research career at JNCSR Bangalore, where he worked from 2007 to 2010. In 2011, he moved to Germany to pursue his PhD from the Technical uh, University of Dresden, and he received the PhD in 2015. And during this time, he had developed some pipelines for discovering small molecule inhibitors against cancer, and he had even received a US patent for his discoveries at that time. And uh, today, he's a research group leader at the Institute of Molecular Immunology at the TM University Hospital, Technical University of Munich. And he leads a dynamic lab working at the forefront of systems biology and computational oncology. And he's currently working on studying the molecular mechanisms behind hepatitis B virus and cancers like glioblastoma, and also on global threats like the SARS-CoV-2. He has over 30 high impact publications in top journals, and he is here to give us a talk on today's topic would be one code, many diseases, decoding HBV, GBM, and SARS-CoV-2 for translational purposes through a computational lens. That's a very, very, very interesting topic. I'm sure that all the audience here are going to be benefited by that. So I welcome you, sir, again, Sainitin. Thank you so much for being here. We are all very eager to hear you. Thank you so much.
Thank you, Abhinand.
Thank you, sir, for the introduction.
Yeah.
Just a minute, sir. Let me preface the meeting by saying that e-certificates will be provided to all participants who fill out the feedback form. The link will be posted at the end of the meeting. So kindly stay until the end if you would like to receive your certificates. Over to the guest speaker. Welcome, sir. Please carry on with the presentation.
Okay, thank you for the kind introduction, Abhinand. Yes, it is my pleasure to present all my recent work where I have used computational biology approaches to decode various diseases. So I work in University Hospital in Technical University of Munich, Germany, in the institute called Molecular Immunology and Experimental Oncology. So in our institute, we focus on mostly on in vivo biology, but where I help to understand these biological experiments and decode omics datasets, which are generated in the lab every day. So we essentially focus on HBV virus infection to understand the molecular mechanisms of immune system in liver. And also, I also work on cancer-related projects using computational methods. So throughout this presentation, I will talk about 4 different diseases: SARS-CoV-2, Cholangiocarcinoma, GBM, and HBV, using computational approaches. How did we decode molecular mechanisms and targets which can help us to identify, to translate into the clinical side eventually? So it all began with how does this bioinformatics approaches came into the picture. It all began with Human Genome Project, which was commissioned in 1993, and it ran over 11 years by collaborating with worldwide institutions, where the aim was to identify, map the human genomes, meaning map the whole human, all the human genes in our mammalian systems. And these drafts have been published in high impact journals like Nature and Science. So during this time, people realized traditional biological methods is not enough to decode these humongous data sets. So that's where the bioinformatics came into the picture. And then that was the beginning of revolution of omics technologies. So what is omics? Omics is used to quantify and characterize various biological molecules in the cells. And this gave rise to omics technologies like microarray methods, bulk RNA sequencing, single cell sequencing, proteomics, and phosphoproteomics, and spatial omics. So all these methods are have been developed throughout the decades, and which has helped us immensely to identify biological processes in various disease settings. So as we all know that biology is a very complex process. And to understand these processes, we need high dimensional approaches to understand the genes and proteins and phosphoproteins and so on. So the omic data sets are generated in labs every day and stored in various databases. And it amounts up to petabytes of data. And the manual inspection of these genes and proteins will take eternity. It is almost impossible to decode all these genes manually or proteins. So how do we analyze these data sets? The solution is bioinformatics. So most of you know bioinformatics immensely helps. And so the next slide, from next slide onwards, I will talk about various diseases where I have used immensely the computational approaches to identify the molecular mechanisms and targets which can be used as drug targets. So let's start with SARS-CoV-2. SARS-CoV-2, we all remember this. In late 2019, we came to know about this, and 2020, 2021, 2022, it has infected worldwide millions of people and disrupted and changed our lives forever, right? And during this time, bioinformatics immensely helped to identify, can you, full screen. Can you see the screen? Sorry for the disturbance. One second, I'll share that again. Something happened. Right. Yeah, okay. So yeah, bioinformatics helped a lot. And so let's look into it. One project which I did in 2020, where we were able to identify drug molecules and a specific target protein. So how does the SARS-CoV-2 bind to the human cell receptors? SARS-CoV-2 has this protein called spike, and it has two subunits, S1 and S2. S1 subunit is the receptor binding domain, which is responsible for virus entry. And S2 subunit is responsible for virus fusion to the membrane of host cells. So the research question was as follows. How can we block virus entering the host cells? So we need to modulate the protein structure of protein structure of receptor binding domain sequence of SARS-CoV-2. So following approach was used, starting with S1 subunit spike protein sequence. And using this sequence, we were able to model the RBD protein structure based on the experimentally available structure. And then we looked into the drug repurposing approach, where we were looked into the binding site similarities between the proteins and this S1 subunit RBD. And then in step 3, we performed molecular docking assays to identify the binding affinities between the drugs and this protein structure. And we also looked into the molecular dynamic simulations of this drug protein complexes. So I will show you the glimpse of the results. So it all started with the protein sequence of spike. And then we looked into specifically into the RBD protein sequence, receptor binding domain sequence of spike proteins from SARS-CoV-2. And then we also looked at the experimental protein structures which were available at that time. Unfortunately, there was only partial protein structure which were experimentally proven at that time in 2020. But the full-length sequence ranged from 13th amino acids to 685 amino acids. So there were residues which were missing from the experimental structure. So this demanded a computational model of full-length structure of RBD protein sequence from spike protein. So we used Swiss model software to predict the 3D structured protein model of S1 RBD, and which shows that it composed of various loop regions, beta sheets, and also the ACE2 binding site region of human, which is the responsible binding region to interact with the human receptor in host cells. And then we looked into because it is an experiment, it is a computational model, and there was no AlphaFold at that time. So we were relying on the computational model, and then we compared this protein structure with already available protein structures derived from X-ray or NMR experiments. And as you see in this here, the black dot shows that this protein structure is in the range of X-ray protein structures. So it is very reliable. And then the Ramachandra plot was visualized to identify favored and allowed regions. And most of them are in favored regions. Most of the amino acids were in favored regions and allowed regions. None were left in unfavorable regions. And then we also compared this protein structure model with partially available structure, and it showed excellent overlap. So showing the confidence that this protein structure model was very reliable. So we took this protein structure for further analysis. So the next step was to perform drug repositioning. So what is drug repositioning? We can use the old drug and give it a new purpose to fight another disease. How does one drug binds to multiple proteins? Because here we are talking about spike protein from the virus. The reason is there will be a similarity between the drug binding sites between the protein structures. So we systematically screened these similarities using one algorithm called drug reposer algorithm, where we compared binding sites of drug binding sites of S1 subunit RBD protein structure, and also structurally resolved of which encompassed 6,000 odd protein structures which are related to the drug binding sites, and then computed the similarity, structural similarity between these binding sites. And as you see in this histogram here, we identified several binding site similarities between the S1 subunit RBD and other protein structures. And then we looked into the top 3 drugs which showed high binding affinity based on the docking experiment. This chlorambucil, tigecycline, and montelukast, which was strongly binding to top 3 drug molecules binding to the S1 subunit. So we took this drug and visualized the binding sites. So the chlorambucil is binding here in the RBD protein, and tigecycline is close to the ACE2 region, and then montelukast was also binding to very close to the ACE2 binding site, which is responsible for human cell interaction. So the docking reveal, so this S1 subunit druggable binds are responsible for drug bindings. But the question is, this is a static approach where we could not see any water molecules or in vivo situations, so that we can accurately predict the dynamics of the protein structure when the drug is bound to this sequence. So to understand the dynamics of the protein structure, we performed molecular dynamic simulations. Using GROMACS software, we prepared the protein structure, and drugs were prepared using the Swiss param software. And then we ran 100 nanosecond simulations using GROMACS software, using a GPU-based computer, which ran for over 5 days for each simulation. And we assessed this output from the simulation data in various levels. First, we looked at the backbone structural stability, where we clearly see that montelukast is disrupting the protein structure significantly when compared to the other drugs, and also compared to the control, where there is no drug bound to the protein structure. And then we assessed the protein compactness. And here also, montelukast is disturbing the protein compactness when it is bound to the protein of RBD. So this clearly shows that montelukast is very vital, very interesting drug, which can be taken further for the experimental use to understand its binding affinities in infected cells. So to do that, we collaborated with the Virology Institute in Munich where they have performed in vitro experiments to understand the binding between the Montelukast and S1 subunit. And indeed, they saw that this particular drug strongly binds to the protein structure. And then it also reduces IL-6 expression in infected blood cells. So in conclusion of this story, so as you saw, computer science, like especially bioinformatics field, has really helped us to identify large-scale drug screening methods. And then we were able to identify pinpoint one drug, important drug, and it also shown experimentally it binds to the SARS-CoV-2 RBD domain, and it also has an effect on immune system by reducing the IL-6 cytokine in infected cells. So we started with the protein sequence, structured modeling, and dynamics, and docking, and so on. And we were able to publish this data in 2022 in Computational Structural Biotechnology Journal. And now let's flip to another disease where we have also used again bioinformatics heavily to identify the cancer drug targets. So the cancer phenotype here we studied was cholangiocarcinoma. So what is cholangiocarcinoma? It is the second most frequent primary liver cancer than hepatocellular carcinoma, other than that. And it is highly aggressive in nature. It seemed rare in the beginning, but constantly the studies show that it is actually becoming more frequent in European countries and the Western world. And it is very highly aggressive in nature, and it is super resistant to the treatment strategies. So now the question is, how can we overcome this resistance in the treatments in the clinics? So we focused on one special set of proteins called chaperones. So chaperones have which also includes heat shock proteins and chaperone activity proteins, which encompass 200 plus odd genes, and they mainly help the proteins to stabilize damaged proteins, and assist in protein folding. And then it also promotes cancer cells to survive and promote the drug resistance. How? Because when patients undergo chemotherapy, cancer cells undergo stress, and these chaperone proteins and heat shock proteins come into the play. They think that they should help these proteins because they are damaged, and they protect the proteins. And then the drug resistance is in the play in the clinics. So these drug targets can be used, these chaperones can be used as a drug targets in cancer treatment. So this study has been performed by my PhD student Manju NJ, and we designed following approach, in silico approach. So we created an atlas of chaperones and heat shock proteins first by curating the literature. And then we looked into the expression of these chaperone proteins in various cholangiocarcinoma transcriptomic proteomic datasets, publicly available datasets. And then we looked into the protein-protein interaction networks of chaperones, and then performed signaling pathway enrichment analysis. And then we have performed an statistical analysis of this network to identify the key therapeutic targets for cholangiocarcinoma, and did molecular docking and dynamic simulations. And then we were able to identify new drugs for the new targets for cholangiocarcinoma. So this is the first of its kind study. Nobody has really done in focused on chaperones, and that too in cholangiocarcinoma. So it all started with literature curation of chaperones proteins. So the first paper we used was published in 2023, where they had 731 chaperone-related proteins. And the second study was from Nature Communications. There were 10 proteins. And then one more paper from 2014 published in Cell Reports. They had 113 proteins. So we overlapped all these chaperone atlases into, as you see in this Venn diagram here. And we took the common chaperones which were published in all these three studies. So this gave rise to 220 chaperones. So these 220 chaperones were further analyzed by using a data set published in the Cancer Genome Atlas, which stores cancer-related transcriptomic data, where we have downloaded the cholangiocarcinoma dataset, which was a small dataset. Number of patients were only 36. but we still looked into that. And then we analyzed the expression patterns of these 200, and we overlapped these 220 proteins of chaperones. And then we saw that 120 chaperones, 22 chaperones were upregulated in cholangiocarcinoma when compared to the normal samples. And interestingly, in 2021, there was a Cancer Cell paper with large cohort of cholangiocarcinoma patient dataset, which was published from Fudan University in China. We were able to download this dataset, which comprised of 262 patients, and they had multi-omic dataset, meaning transcriptomics, proteomics, and phosphoproteomics. So they had a common list of proteins. So we used this list of proteins and then compared to the 220 chaperones. And we found 18 chaperones. So these 122 chaperones from TCGA comparison and 18 chaperones from multi-omic dataset from large cohort of cholangiocarcinoma dataset was further compared, and this comparison gave rise to 8 chaperones which were commonly found in both these studies. So there were heat shock proteins and there were other chaperone-related proteins. So these 8 chaperones were further analyzed using protein-protein interaction network, where we constructed protein-protein interaction network using the multi-omics dataset from large patient cohort and used statistical methods to identify the key connecting molecules of chaperones and then perform the pathway enrichment analysis of this hub-related, meaning the key proteins which are related to chaperone activity. And here you see in this scatter plot, as I said in just now, the statistical analysis, which we used something called degree and betweenness. So in simple terms, degree means the number of interactions of proteins in the protein-protein interaction network. The betweenness meaning high betweenness, showing that that particular protein is very important in the signaling network. And then we also looked into one more parameter called coreness of the network, meaning that particular protein has high coreness value. Then if you remove that protein, the whole network is disseminated. So as you see in this bar plot here, we have plotted that, and also the scatter plot. When you look at closely to both of them, these two proteins stood out. CRY-AB and Pre4HA2 showed high values of degree, betweenness, and coreness. So out of these 8 proteins, we came down to 2 proteins, which are important for upregulation and also playing a major role in cholangiocarcinoma setting. And they are playing a role in chaperone activity. So we used this particular 2 proteins, and then we downloaded FDA approved and various institutions approved cancer related drugs. And then this CRY-AB and P4HA2 protein structure were experimentally derived. So we used protein data bank and then predicted the druggable binding sites of these 2 proteins, and then performed molecular docking analysis of these 280 drugs which are FDA approved and EMA approved. And then looked into the top 5 drugs which showed highest binding affinity. For CRY-AB, we found 2 drugs, tucatinib and parsenitinib, which showed similar binding affinity. So we took these 2 drugs and then visualized this binding pockets. As you clearly see here, the binding pocket is interacting with this particular protein binding site with the drug by van der Waals interaction, conventional hydrogen bonds, and pi-pi interactions, and so on. So the second protein was, for the CRY-AB, these are the 2 drugs, best hit drug molecules. And for P4HA2, again, the top 5 drugs were looked into, and then we saw that regorafenib is stood out for with highest binding affinity. And we used this particular drug for further analysis to we looked into the interactions between the drug and the binding pocket, which is druggable. And here also we see that there are van der Waals interactions between the drug and the binding site, and there is hydrogen bonds and pi-pi stacking interactions. So regorafenib was the best hit for the P4HA2. Again, this is still docking. We also looked into dynamics again to understand deeper understanding of these interactions of drug and the proteins. So as you see here, this CRY-AB had 2 drugs, tucatinib and parsenitinib, but parsenitinib did not show significant results from the MD simulations, so this was discarded. So we took tucatinib because it showed clear significant differences, where we saw that this particular drug, when it is bound to the cryAB, it is disturbing the backbone and also causing the disturbance in binding pockets. As you see in this box plot here, when there is no drug, there is no much difference, but when the drug is bound to the binding site of cryAB, there is significant change. And the similar analysis was performed for P4HA2 and regorafenib. And here also we see that the drug affects the backbone of the protein and also the binding site at the amino acid level. And then we also looked into the binding assay using the molecular trajectories. There we clearly saw that drugs strongly bind to these two protein structures. And in conclusion, in cholangiocarcinoma, these two chaperones are highly upregulated and strongly connected with various tumor-related pathways like EMT transition for P4HA2, P53 signaling, MAP kinase signaling, and for cryAB, it is EMT transition, epithelial mesenchymal transition, angiogenesis, glycolysis, and hypoxia, and various signaling processes. So we were able to propose two drugs against these two proteins. So this needs further experimental validation, but this was the first study in cholangiocarcinoma where my PhD student has rigorously looked into it, and then we were able to identify novel molecules which are responsible for CCA resistance in therapy, and also the drug molecules which can be used in future. And we were able to publish this data in 2023 in Computational Biology and Medicine. So these were two studies. So until now we had SARS-CoV-2, liver cancer, and now let's move to another cancer phenotype called brain cancer, glioblastoma multiforme. What is glioblastoma multiforme? It is a primary brain tumor and highly aggressive in nature. This particular brain cancer arises from glial cell types like astrocytes, oligodendrocytes, and microglia. And then the highly aggressive immune microenvironment also helps these glial cells to promote the tumor in the brain. So this particular tumor phenotype is very heterogeneous because it has various cell types involvement like astrocytes, microglia, oligodendrocytes that further divided into 3 cell types: oligodendrocyte precursor cells, newly formed oligodendrocytes, and myelinating oligodendrocytes. And the tumor microenvironment, which is related to the immune system and stromal cells, also contribute heavily to the treatment resistance and tumor occurrence. So this microenvironment promotes tumor progression and also contributes to the poor prognosis of the patients. So the question we asked here is, what are the tumor microenvironment interactions involved in glial cells, and what are the molecules which are interacting with the tumor cells, I mean the immune cells and stromal cells, like focused on tumor microenvironment in GBM. So to understand this process, we have designed along with my student Sai Krishna and Alagamai, we have designed a computational approach where we have looked into the published RNA sequencing transcriptomic dataset from brain cancer using TCGA database. We have downloaded that, and then we have reanalyzed. We divided the patient samples into the high and low scoring immune patients and high and low scoring stromal patients. And then we also looked into the differential expression between the normal and between the high and low immune and stromal cells patients. And then we looked into the co-expression networks, and then we mapped these co-expression networks into the glial cell type enrichment. And finally, we looked into the prognostic analysis to identify the prognostic biomarker, which is responsible for tumor microenvironment in brain cancer, and then validated these results using the single cell RNA sequencing patient dataset and correlated the expression of that particular gene with immune cells and stromal cells, which is in relation to tumor microenvironment. So first, as you see in this bar plot here, we see that there is a significant upregulation of genes in immune cells and stromal cells patients, like 793 and 733 genes. And the pathway analysis has clearly shows that there are various tumor-related pathways are regulated up or down in immune and stromal cells in GBM patients. But we all know that genes doesn't work in isolation. They work in networks and give functional output. Now the question is, how do we connect the genes in immune and stromal cells from this GBM dataset, which were identified the differential expression like 793 and 95 and so on. To answer that, we have used an algorithm called weighted gene co-expression network algorithm to identify modules, connected gene co-expression modules in stromal cells and immune cells from GBM expression datasets. As you see in this dot plot here, we downloaded the marker gene sets of glial cells, meaning for astrocytes, there are some marker genes for microglia and for also the oligodendrocytes. We overlapped those genes from the literature onto this module genes and then computed the enrichment. And if you see closely, that microglia is significantly enriched in module number 6, and NFO is significantly enriched for module number 6 also. And then we also looked into the highly connected modules. And again, we saw microglia and also NFO and some OPC highly connected modules in immune cells dataset. Similar analysis was performed for stromal cells. Here also we saw microglia and oligodendrocyte enrichment. So overall, this co-expression network analysis highlighted that glial cells are enriched in this particular network modules in immune cells and stromal cells. It still gives a large number of genes. So we need to identify key connected genes from these networks. So we used an approach called hub gene identification and computed intramodular connectivity of each gene in these particular modules of glial cell type enriched modules for immune cells and also the stromal cells. So if you closely look at it, there are some genes which are highlighted in bold. So these genes having the highest score mathematically in this particular network. So we say that, okay, these genes are important with high score of hub scores. So we took these genes for the prognostic analysis further to identify the survivals, how these genes are responsible for survival of the patient from GBM prognosis. We performed a univariate analysis first and looked into these genes which are highly connected, and we were able to identify some set of genes like ITGAM, LCK1, ITGB2, THBS1, and so on, which are significantly related with survival. By combining the univariate and multivariate analysis of survival data, we were able to pinpoint to set of common genes which are responsible for bad or good prognosis. Meaning, if that particular gene has high expression, patients are dying faster, or if that particular gene is having high expression, then it is patients are surviving longer. So good or bad prognostic markers. So using this set of genes, we further, we wanted to look into the precision medicine approach, meaning we had until now the bulk RNA sequencing data, meaning the pool of cells. But if we want to precisely target a gene in any disease setting, we need to look at the single cell level. So there is a technology in omics called single cell RNA sequencing approach, where people isolate the cells individually and sequence the genome in each cell. And then we can visualize it as UMAP, as you see here. And then we can also annotate these clusters from this data to glial cells. We were able to identify all the glial cells here. And then when we looked into the expression of these prognostically relevant markers, which we identified in the previous analysis, especially ITGB2 stood out because it is highly upregulated in both immune cells and stromal cells. And there we saw also in bulk RNA sequencing that it is upregulated, and also in single cell level, it is upregulated in microglia module and also the newly formed oligodendrocytes. So we took this particular gene for further analysis. And we also looked into the ITGB2 signaling network, how this is communicating between the glial cells like microglia and NFO. We saw strong communication pathway links between microglia and NFO, which is contributing to the tumor oogenesis. So, and then finally, we also looked into how this particular gene is connected with various genes and what are the molecular pathways which are regulated in stromal cells and immune cells. So this shows that ITGP2 is well connected in various tumor pathways in immune and stromal cells. The upstream transcription factor analysis, because we are talking about transcriptome here. So when we talk about transcriptome, the transcription factors regulate the transcriptome. So we wanted to identify which transcription factor is responsible for this particular gene. So we found IKZF1, which regulates ITGP2 gene. And the main aim of the project, at the beginning I told you that we wanted to look at the gene expression and correlation with the tumor microenvironment from immune cells and stromal cells. To do that, we have computed the expression correlation analysis between the immune cells, and we found significant correlations of this ITGP2 expression with myeloid dendritic cells, macrophages, monocytes, and B cells, and neutrophils, and so on. And at the stromal cells level, this particular gene expression is well correlated with fibroblasts and adipocytes. So this shows that tumor microenvironment indeed interacts with ITGP2. And when the ITGP2 expression is high, the patients are dying faster, according to our survival analysis. So in conclusion, by using bioinformatics methods, we were able to delineate the mechanisms of tumor microenvironment in GBM patients using the bulk RNA sequencing transcriptomic dataset and also the single cell level RNA sequencing dataset. This highlighted again ITGP2 and IKZF1 transcription factor, which regulates various immune-related pathways and tumor-related pathways like VEGF signaling pathway, EGFR signaling pathway, and tumor proliferation. And then we were able to publish this data in 2023 in Computational Biology and Medicine Journal. So now we enter the last disease. How here, where we have combined immensely the biological experiments and also the computational experiments together. So until now, I showed only mostly bioinformatics approaches, but in the next project, which I will show you how bioinformatics has really helped to pinpoint molecular mechanism of CD8 T cell function in HBV patients. So what is HBV? HBV, it is a hepatitis B virus. It infects primarily liver. And the chronically infected HBV patients are over the year, like there are 300 million patients are reported, and over 1 million people die every year. Why? Because HBV specific CD8 T cells are very rare, because immune system should protect us, but immune system doesn't work because CD8 T cells are dysfunctional in chronically infected patients. When patient is infected with acute infection, our immune response really works very well. And over time, in few days, we were able to clear the infection. But in chronic infection, immune system doesn't work. Why? CD8 T cells are dysfunctional. So the question here, in the main question of this project was, why CD8 T cells are dysfunctional during chronic infected patients? So to answer this question, so this was done in collaboration with Immunology lab in our institute started with Miriam and Jörg Wolliber, who are the collaborators of this project, where they started with preclinical mouse model system called adenovirus-based system, where they have acute infection and chronic infection. And the experiment shows that when the acute infected mouse model was able to clear the infection very fast in 20 days, but chronically infected mouse model doesn't clear. It stays over the period of time. Because, as I said, CD8 T cells are dysfunctional. So to understand why, how come this, what is the regulation happening in CD8 T cell dysfunctional state in mouse models, we performed bulk RNA sequencing experiment using acute and chronic CD8 T cells. And then I performed bioinformatics analysis to identify the differential expressed genes in chronic infection and in comparison to acute and chronic. And I was able to identify the transcription factor which is responsible for CD8 T cell function. Here is the network. So here we saw that CREM transcription factor is hallmark of this chronically infected mouse model system, which is regulating various genes in CD8 T cells. And CREM is also regulator of CAMP signaling pathway, which is well known in the literature. But this is for the first time we saw CREM transcription factor is responsible for chronic CD8 T cell function. So here, this was preclinical model. It is a lab virus. What about, can we identify the same gene in HBV system? So to do that, we collaborated with Roche pharmaceutical company, where they had HBV mouse model, and they performed RNA sequencing analysis. And then we also looked into we compared this HBV mouse models from acute resolved infection and persistent chronic infection. Here we saw the clear, there are differences between these expression systems according to the principal component analysis. And we also looked into which transcription factors are interesting. And one of them, again, CREM. So the prediction which I made in preclinical model came true again in HBV mouse model from the independent system. So CREM is really important here, and which is regulator of CAMP signaling pathway. So until now, okay, still mouse model. What about real patients? Can we find CREM signature, CREM-related target genes in patients of chronically infected HBV-infected patients? So we started with 3 patients. That was dataset 1, where we performed single-cell RNA sequencing of CD8 T cells, of HBV core-specific CD8 T cells. Here also we saw, again, along with other transcription factors, significantly the CREM signature is enriched in each patient. As you see in this box plot here, when we compare to the low expression and high expression, this particular CREM signature is high in each patient. But it's only 3 patients. So we also independently validated in another patient dataset with 11 patients, where we clearly see that CREM targets, like CREM, when there is a hepatitis B infection, it is upregulated. When the patients are cured, there is no expression of CREM. Meaning, this particular CREM transcription factor is really vital for translation purpose. Again, this was identified by bioinformatics approach. Using traditional looking at the Excel sheet approach, we will not be able to find this. So using sophisticated mathematical approach, using bioinformatics, we were able to pinpoint this target gene and showed that in various levels, it is important in mouse and HBV and patient infected patients. So overall, we found at the end, there were also other experiments. Due to the time crunch, I'm not able to show this, but you can look at the paper later. Here we clearly see that CAMP signaling pathway, which is regulated by CREM, is making the CD8 T cells dysfunctional. And the liver tissue cells are interacting with the virus-specific CD8 T cell receptors. And this mechanism delineation will help in future to look into the silencing approaches of HBV-specific CD8 T cells after the therapeutic vaccination for the patients. And we were able to publish this data in Nature in a high impact journal in 2024. So from all these studies, I hope I was able to convince you these years, in last 15 years or so, how bioinformatics advancements have immensely helped in various disease-related projects. And we were able to identify novel mechanisms, novel targets, which were not possible so easily before. I would not say it is easy till now, but it will really reduce the timelines. Because as I said in the beginning of my presentation, manual inspection of these genes from large omics data sets will take a lot of time. It will make us, it is impossible to find novel insights, because people were always looking into the already known genes. If they find, they're happy. But I ask a question, can we identify novel stuff? We had so much computational power now, and using this bioinformatics advancements, we can identify novel related proteins and genes which can be used later in translational approaches. Of course, right now the cutting edge is generative AI, like which uses various algorithms called variational autoencoder algorithm, transformer language models to identify novel compound libraries by getting trained on the known compounds and predict the novel compounds which doesn't exist. So that is like a cutting edge in the bioinformatics right now. And this brings me to the conclusion. Of course, all this work is not possible without fantastic team of students and collaborators. So the SARS-CoV-2 was started in 2020 until 2022. This was done by a PhD student, Max Ludemann, and a collaborator, Daniela Stadler from Biology Institute. And Cholangiocarcinoma study was performed by my PhD student, but from BITS Pilani, Dubai, Manju NJ. And my master students were immensely very dedicated to work in glioblastoma projects, Sai Krishna and Alagamai, and my long-term collaborator, Associate Professor Swati from MS Ramaya University, India. And the HBV project started in 2016 and ran until 2024. This was an immense effort from the lab where I work in the institute, from PhD students and postdocs like Miriam and Nina, and my director of the institute, Professor Persik Nole. And this project we were able to collaborate with pan-world related collaborators, like from Technical University of Germany, Heidelberg University, Germany, Roche Pharma from Switzerland, Harvard University, and ETH Zurich, and Cambridge University from London. And thank you for attention. And of course, I'm open for collaborations, and here is my contact.
Thank you so much, Dr. Nitin. I believe we sent you a questions from a document.
Yes, I do have. Yeah. I do have this question and answers. Yes.
Yes. So before we could move on to the Q&A session, can we address those first, please?
Yes. Sure.
Thank you so much.
I hope everybody was able to hear my voice clearly. Yes?
Yes, sir. We heard it clearly and perfect.
Okay. Yeah. So, I received some questions. They were brilliant questions. I can really admire the passion of the students right now. At this age, they were able to find new questions to challenge various research. Yeah, it's very interesting. But the first question is related to nanotechnology from Sakshi Pandey. I don't work in nanotechnology. I work in bioinformatics, but I try to answer this. But I think this requires maybe reframe or further analysis or whatever. As I said in my talk, we can use multi-omics data sets, which identifies various enzymes, molecules, and various microenvironment related genes, pathways, and so on. From these multi-omics datasets, I believe one can really look into the nanotechnology approaches to map these various ligands and predict the triggers of these biological pathways, which vary from tissue to tissue, disease to disease. But we need to learn all these multi-omics datasets and then design a nanotechnology-related approach to combat the disease, I hope. So let's see. Maybe I think people are working on it. So another question from Sami from BS Biotechnology. So he asked that how can AI will help to handle the rare cancers like cholangiocarcinoma? As I said, cholangiocarcinoma, it seemed rare, but these days it is becoming more prevalent. Yeah, but of course there are rare cancers. But using these rare cancers, one can use various sophisticated artificial intelligence models like transfer learning by learning the data from pan-cancer patient datasets from TCGA or published datasets, and then identify the module-related pathways, networks and pathways rather than single genes. I think this kind of approaches will help to train and predict the novel pathways or molecules from rare cancer phenotypes. That is a very good question. Yeah. So the next question is again about cholangiocarcinoma. So what is the relationship between the viral entry and replication with cholangiocarcinoma? But cholangiocarcinoma is not related to really the virus, because the main trigger of this cholangiocarcinoma is from chronic hepatitis. The hepatitis B virus leads to HCC and also eventually to cholangiocarcinoma sometimes. So this involves various pathways like inflammation, fibrosis, DNA integration, and various immune-related pathways. So by studying these mechanisms, one can, of course, look into the molecular targets. As I stated in my talk, I focused on, me and my PhD students focused on chaperone activity, but of course, this can be also improved and extended into how these chaperones are linked with immune system or tumor microenvironment of cholangiocarcinoma. That is one way to go about it. So the second is another question from Prasad from Berhampur University. The translation is the unit, okay, different mechanisms, how translation application of discovery, addressing the unit, okay, from the various tissues. So we need to design computational approaches. Of course, there are already context aware, meaning the disease specific approaches, like using artificial intelligence. We can look into predict various factors like enzyme activity and in different organs, in receptor profiles, in different tissues like lung, brain, liver, and by context, by meaning the tissue awareness, like spatial omics from various organs will help to identify molecular targets and molecular insight pathways from organ specificity to efficacy. Yeah? And Karun from Ramachandra University. So how can single code can help to decode various diseases? I mean, of course, there is no absolutely single code, but one can design, because various diseases have sometimes mostly have shared molecular space, meaning they might have a shared pathways from brain to liver, because there is this gut-liver, gut-brain axis method where people study heavily on Alzheimer's disease, how the interactions happens between the gut and brain. So that kind of studies can be performed and looked into the shared modules, all these publicly available data sets from omics methods. We can look into the how these molecules are connected and maybe working on a common pathway, and maybe we can delineate that and then look into the unique and common pathways. So that is one way to go about it. So how this in silico work will get an equal recognition as wet lab work? Okay, Palak, Raghav, it's a very good question. I used to think the same, but the institute where I work now immensely relies on bioinformatics. Although they work in every day in the lab, in vivo biology, mouse models, and so on, and they do the omics technology approaches, and then they come to me. So without bioinformatics, as I showed the Nature paper, which we published recently in Nature from the HBV research, which was hand in hand, meaning they did the experiment and then it came back to me, and then I predicted one molecule and they went back to the wet lab again. So it goes in hand. So there is no, because if you do everything dry lab, it always stays in the computer. But eventually the main goal of all our research from bioinformatics, from wet lab, we need to translate into the clinics, because we need to give medicine to the patients eventually, right? So to do that, we need both. We cannot rely only on one. Only on one will really put us in a very dangerous position, because sometimes we only on the wet lab scientists will rely only on the known stuff, but dry lab scientists will rely only on the novel stuff. But dry lab people will think that that is novel, but is it really novel? That needs to go to the wet lab again. So it goes hand in hand, yes? Then we can make groundbreaking and life-changing discoveries. I remember a few years ago, the molecular dynamic simulation approach has got Nobel Prize, and a couple of years ago, AlphaFold got a Nobel Prize. So you can see. So the AI and the computer science is getting equal recognition as wet lab work, but both should go in hand though. So another question from Sahiti from Ramaya University: how the AI agents will help in biotech sectors? Yeah, of course, AI agents is a current trend now. I saw a Nature paper a few days ago where they have used AI agents to identify antibodies, novel antibodies, from checking the literature to look into the networks, how they are interacting, and then they went into the lab. So it started from AI agents to the lab. So they were able to identify novel antibodies and for SARS-CoV-2. So this kind of studies will help. So I think there are some few people in the world, people are working on this kind of AI agent technologies, but not really immensely yet. But more related to IT, I mean, pure computer science people are working on it. So let's see how it really helps. But there are some very nice clues that AI agents will help very nicely to identify the drug targets. Yeah. So how does the spatial omics? Yeah, that's a very good question too. Spatial omics is a current trending technology. In our lab, we just started in the institute. The new data is just started coming in a couple of weeks ago in HBV research. So where we try to identify cell types, liver cell types, and look into the receptor and ligand interactions. The spatial technology essentially meaning to identify the RNA and proteins where they are located in the tissue. So the tissue specificity. And using this technology, we can really understand what is happening by combining the single cell and spatial technology. We can really identify the novel molecular mechanisms in various diseases, of course. So this is like current methods, very new, recently getting developed, and people are using it. So let's see how far it goes. But it requires large-scale computing. Again, with only the wet lab approach doesn't help here. And the last question was research opportunities in Germany for Indian students, how to bridge skill gap in industry and academia. Okay, so there are pharmaceutical industries in Switzerland, in Germany, all over Europe. They offer PhD positions in pharmacy industry, where you can learn, collaborate with research universities, and also how industry works. But if you do PhD in a research university and go to a postdoctoral fellowship in the industry, so that will help you to understand how industry works and how academics works. So we are also like, there is one big project which we did last 5 years. We were running this. So recently we collaborated with one pharma industry to identify novel drug molecules against that particular protein. So by this, we also learn, we share the data and we learn how the industry works. But if you want to learn personally, I would suggest to do PhD in company, where you have a chance to get experience in real time, how the industry works.
Thank you so much, Dr. Nitin. I believe you have a question in the chat box from...
Yeah, I'm looking at it. Yeah, first question, can we design reverse engineering? Yeah, there is an algorithm called genetic algorithms, which have been used for protein sequencing analysis from long time. And they have been working on it. Yes, we can design, and by using, especially combining with artificial intelligence, will help to identify the mutations, cancer mutations, and so on. Yes.
If there are any more questions from the participants, please type them out in the chat box, or you can turn on your mics and directly ask the questions, please.
I believe we have no more questions. Thank you, everyone. Thank you, Dr. Sainitin, for the very insightful lecture.